on this her sophomore year. She's a junior now, so she's been working on it for a while, although she did take an extended break last year when she was doing iGEM instead, the synthetic biology competition. And so she's going to tell you about what she's been doing. Some of you may have heard about this topic before because my PhD student, Adriana Bingham, worked on a within-host model of polio. So Wei is extending the model that Adriana did. And this is in collaboration with Simone Bianco at IBM. So yeah, Wei, you can go ahead and get started.
Can everybody see my screen?
Yes.
Yeah, so nice to see everyone here. So today I'm going to talk about the within-host model of poliovirus control by defective interfering particles and interferon. Before talking about math, I think it would be helpful to give some biological background. So our main focus of the model poliovirus is a positive sense single-stranded RNA enterovirus. And poliovirus usually begins in the intestines and travels to other parts of the body. And poliovirus is most destructive in the central nervous system. And it is known to cause poliomyelitis. So the major problem associated with poliovirus is that although an oral vaccination was created, the virus is not yet eradicated. And that's the major motivation for us to build our model. And with that in mind, some scientists have proposed a possible treatment strategy, which is using defective interfering particles. You may have heard about that before, but just in case, DIPs are non-viable variants of wild-type virus, so they are unable to complete the replication cycle by themselves due to the lack of the capsid protein. So they act sort of as a parasite of the wild-type. Specifically, during the co-infection with the wild-type, DIP steals capsid protein from the wild-type, thereby reducing the overall wild-type viral load. And as the figure indicated here, DIPs usually have smaller genome because they don't have the genome coding for the capsid protein. So DIPs are able to replicate faster and thus outcompete the wild-type. And once the parent virus population has been depleted, DIPs are incapable of reproducing by themselves further, so they also gradually go away themselves. So we are building the model in order to assess the feasibility of using DIPs to control the wild types. And specifically, we also want to know when to introduce DIPs and also how much DIPs should we introduce initially to maximize the effect. So this is the model that Dr. Adriana Bingham built before in her preprint. So this is the flow chart of her model. So there are 6 variables we are focusing on: the susceptible cells and 3 types of infected cells, which are those infected by wild-type only IW, or those infected by DIPs only ID, and those co-infected by DIPs and wild-types IDW, and also 2 types of virus, wild-type and its variant DIPs. So susceptible cells have constant birth and death rate, and they can be either infected by a wild-type virus at rate K or by DIPs at rate K. Notice that we cannot successfully infect a cell with only 1 viral particle. For wild-type, we need n amount of viral particles, and for DIPs, we need gamma times n number of DIPs. to trigger the successful infection. And wild-type virus are able to lyse the IW cells by themselves at rate delta and thus release delta times an out amount of wild-type viral particles. This indicates the process of viral replication. As a contrast, you can see that ID don't have the cell lysis reaction. That's because DIPs cannot do viral replication themselves due to the lack of capsid protein. The only way they are able to reproduce more viral particles is through co-infection with wild-type virus. So if ID co-infected with wild-type virus, they become IDW, and they are able to have cell lysis at rate delta, and thus release delta psi and out amount of wild-type or delta psi rho and out amount of DIPs respectively. And also wild-type and DIPs are cleared out at rate C. And this is the model that Adriana built before. And we also have two additional aspects to consider to make the model more approximate to biological reality. So the first aspect is the incorporation of stochasticity. The major motivation for us to account for that is that according to the simulation result from the deterministic model indicated by the figure on the right, so we can see that at the very beginning, the DIPs drop down to an unrealistically small number. This is legitimate for deterministic model, but we are kind of concerned if for a real biological environment where every variables are only allowed to have discrete value, if that means it would actually go down to 0, which means that DIPs just go away at the very beginning and they are not able to actually successfully persist. And we incorporate stochasticity into our model through philosophy algorithm. You may have heard about that before, but just in case, I'm gonna explain it very briefly with a very simple chemical reaction system. So there are 3 possible reactions here: the birth of A, and with 1A and 2B, they can be turned into C at rate K, and also the death of C at rate D. So there are 3 possible reactions here, and they have rate B and rate A, concentration of A times B times K, and concentration of C times D for the reactions to happen respectively. We add them up to get the R total, and for each time step, we randomly generate a number along this line and see if it's actually falls at this interval, we will have the reaction 2 to happen. And correspondingly, A will be decreased by 1 number, B will be decreased by 2, and C will be increased by 1. And so using the GlassPy algorithm, we are able to transform the deterministic model into stochastic model. So in total, there are 11 possible reactions in our model. And as indicated by this line at the bottom, each of them has their own reaction rate. And at each time step, we generate a random number and see which interval it falls to trigger the corresponding reaction. So after incorporating stochasticity and using the same initial value of DIPs, which is equal to 1,000, we found that the DIP quickly dropped down to 0 at the very beginning. So our conclusion is that after taking stochasticity into account, DIPs may not be able to persist. However, after we increased the initial DIPs from 1,000 to 5,000, according to the simulation, the DIPs are able to persist. And the second—
Actually, Wei, can I have you go back to the previous slide? No, the one with the 4 panels. Yeah, just wanted to point out, these are 4 different panels going further and further into time. So what you see on the bottom right is after a very long time, 160 hours, the DIP levels have gone quite high. And so they're persisting in the system and hopefully helping to control the disease there.
Thank you.
Yeah, and actually this would be a really good time for questions if anyone has questions about the stochastic aspects before Wei transitions into talking about the immune response.
Can you explain why there's these sudden jumps in the population of dips and then gradual decline? Gradual decline I can understand, but why the sudden jumps?
Yeah, that's actually a very good question. So the sudden jump of the DIP value actually indicates cell lysis happen. So like for each cell lysis, there are actually about 500 DIPs released. So the sudden jump actually corresponds to 1 cell lysis event, and it falls down also very quickly after that, because if we refer to this, the mechanism of this Gillespie algorithm, we can see that we can see that like reaction 10 here, which corresponds to the co-infection of the IW, or like, yeah, just using that as an example, or like reaction 4 here, the infection of the ID only. So after the cell lysis and the number of DIPs increase, the possibility for R4 or like R10 to happen also get increased. So it is very likely for either of these reactions to happen right after the cell lysis. So, and every infection process actually takes also around 500 DIP particles. So that corresponds to the sudden drop after the sudden increase of the DIP values.
So you are counting dips that are not in the cells, is that right? You
Yes.
said the cell breaks and you suddenly get more, and then they get into a cell, so you would suddenly have less.
Yeah, these are the ones that are free in the bloodstream. This is not including the ones that are inside the cells.
Okay, thank you.
Okay, I guess we can move on to the immune response.
Yeah, the second aspect we are considering is the incorporation of interferon. So interferons are just cytokines used to trigger the protective defenses of immune system, which protect uninfected cells from viral infection. Our major motivation for us to consider that is also because according to the simulation result of the deterministic model, wild type is not completely cleared out at the very end. So we want to know if this is possible with the incorporation of the immune system. Since it is too complicated to include all the elements of the immune system, we simply use interferon as a representative. And this is the model after the incorporation of interferon. Everything marked in green are new variables and parameters introduced as a result. You may notice that there are no DIPs in this model. That's because adding interferon actually introduce already a great amount of parameters, and it's kind of complicated to also take DIPs into account at the same time. So for now, we are just exclude DIPs to avoid things getting too complicated, but we will include them back eventually. And interferon indicated by F can be produced by IW at rate Q. And they have two main functions. The first is to kill the infected cells at rate kappa and thus prohibit the viral replication process. And the second is to transform some susceptible cells to refractory cells. Those are cells that are not able to be infected by virus. But notice that they could not stay at this state forever, and they will actually reverse to susceptible cells at rate R. And during our process of incorporating interferon, we have two major obstacles. The first is that we expect interferon parameters to depend on specific species, virus, and tissue type. But in the literature, it's impossible for us to find parameters correspond to what we want exactly. And secondly, parameters from other literatures are fitted through their models, and their models are different from ours. And for now, we are not planning to solve the first obstacle at this point. But for the second obstacle, we are planning to get the data set of interferon and other associated variables from literature, and then conduct parameter fitting using our model instead of directly borrow the value of parameters from their paper. And that leads to how we have done our parameter fitting and some problems we have encountered. So the method we have been working with for a while is using the SciPy optimized least squares package in Python. This is an example of a typical data set we are able to get from the literature. So usually there are data for more than one variable. For example, here we have both viral load and interferon. So for each type of data, we calculate the predicted value subtract by the true sample value, square it and divide it by 1/10 of the largest sample value in this type of data set. We do this also for all other types of data set, and we sum them up and try to minimize their sum during the fitting process. And there are actually a lot of possible methods for us to choose and also parameter values for us to set in this SciPy package. So it's kind of hard for us to decide which choice can generate the most optimal results. So we use simulated data with parameters we assign, so they are known beforehand. And we try a bunch of possible options. Sorry.
I need to mute somebody. Hold on, let me see who's unmuted and has background noise. Now we're good. Okay, you can go on, Wei.
Yeah, so, and we conducted data fitting with different possible options of the method and parameter choice. And we also increase deviations of the initial guesses from the target value, since the initial guesses also influence the results we can get. So after doing this, we find the best method we have found so far is fitting the logarithmic of parameters instead of the absolute value, and also use the LM method in that package, and take the results from the second round. So basically, after we're done with first round of fitting, we take the results we get as the initial guesses for the second round. And the results we get from the second round is more reliable. So we also have found some problems in this process. So the first one is that we found the fitting results largely depends on how close the initial guesses are to target values. So here are the three graphs of indicating the increasing deviations of the initial guesses from the target value. So the STD equals to 30% here means that initial guesses are randomly generated from a Gaussian distribution with mean value set to be equal to the target values and standard deviation to be 30% of the mean value. So with increasing STD, the deviation of the initial guesses to target value gets increased. So we can see that when STD equals to 30%, the fitting result is still quite decent, but if we increase it to 60%, it's pretty far off from the target time series. And another problem we have encountered is that sometimes fitting results still deviate from the target values, even when perfect initial guesses are given. We are extremely confused by that because when perfect values are given as initial guesses, so the residues actually starts from 0. So theoretically, it shouldn't become any larger than that. So our guess is that probably the algorithm search globally for the optimal parameters, and it probably accidentally gets stuck at a local minimum where it's actually bigger than 0. And another method we are considering to use, but we haven't actually tried that yet, is Latin hypercube sampling. This method allows us to search broadly across the space without trying all possible combinations. So for example, if we are fitting for just 2 parameters, and each point here indicate a possible combination of the value. And if we are done with trying this value over here, we will not try with all possible combinations along this row and this column. And some future works we are planning to do is first to include DIPs in the interference model. For now, we have excluded them for the simplicity. And also, for now in the model, we add the DIPs exactly at the same time when wild-type virus is introduced. That is not possible in reality. So for future work, we want to vary the time to add DIPs later, and to make this more realistic and also find the most optimal choice. And also for the initial value, for now we just have kind of randomly guessed the 5,000 as the DIP's initial value. So it might be like slightly lower than that to also let the DIPs successfully persist and also have more effective treatment. And for now, we only have one patch model, so you may assume the whole flow diagram is only of accounting for reactions happening in one organ. But since poliovirus is known to be able to transmit it from one part of the body to other parts of the body, we eventually may build two patch model. Yeah, that's all of my presentation.